Good morning, everyone. I am pleased to welcome you to today's presentation. Today, we will be exploring the qualifications of a UV visible spectrophotometer. I am Dr. S.K. Singh, and I will guide you through this topic. Over the next few minutes, we'll cover the key aspects of this important instrument, including its features and significance in scientific analysis. Let's get started. Let's take a closer look at the scope of our validation process. This applies to UV visible spectrophotometers used in several critical areas. These include assay, which is essential for measuring the concentration of active ingredients, dissolution testing, to evaluate how quickly a drug releases into the body, content uniformity, ensuring each dosage unit contains the correct amount of active, and finally, identification tests, which confirm the presence of specific compounds. Now, moving on to the objective. Our goal here is to establish documented evidence that the UV visible spectrophotometer performs consistently and accurately, just as it's intended to do. This is vital for maintaining quality and compliance in our testing processes. Next, let's touch on the qualification lifecycle, which is a structured approach to validation. It starts with design qualification, or DQ, where we verify that the instrument design meets our needs. Then comes installation qualification, IQ, ensuring the device is installed correctly. Operational qualification, OQ, tests that the instrument operates properly within specified parameters. And finally, performance qualification, PQ, confirms that the device performs reliably during routine use. Together, these steps help us ensure that our equipment is always up to standard, supporting our overall goal of delivering high-quality, consistent results. Now, let's talk about the installation qualification, or IQ, checks. This is a crucial step to ensure everything is set up correctly from the start. First, we verify that the instrument has been installed according to the manufacturer's recommendations. This means following the specific guidelines to make sure the equipment is positioned and assembled properly. Next, we look at the environmental conditions. It's important that the temperature stays within 20 to 25 degrees Celsius and humidity levels are at or below 60%. These conditions help maintain the instrument's optimal performance and longevity. We also need to check the power supply and earthing. Proper electrical connections and grounding are vital for safety and for preventing any electrical issues that could affect the instrument's operation. Then, we verify the software version and ensure that the correct license is installed. This guarantees that the instrument is running the latest, most secure software and has all necessary permissions. Accessories are also checked at this stage, things like cuvettes, the printer, and cables, to confirm everything is present and in good condition. When all these checks are completed, we move to the IQ acceptance phase, where we verify and document that all components are in place and functioning correctly. This documentation is key to maintaining a clear record of the installation process, confirming that everything meets the required standards. In summary, these steps help us lay a solid foundation for reliable, accurate results throughout the instrument's life cycle. Now, let's take a closer look at operational qualification, or OQ, which is a crucial part of validating our analytical instruments. According to USB 857, there are several key parameters we need to verify during OQ to ensure the instrument is performing reliably. First, we have wavelength accuracy. This means we need to confirm that the instrument measures wavelengths correctly, because precise wavelength measurement is essential for accurate analysis. Next is photometric accuracy, which involves verifying that the instrument's absorbance readings are accurate and consistent, ensuring reliable quantification. Stray light is another critical parameter. It's about checking how much unwanted light reaches the detector, which can interfere with the results. We want to keep this as low as possible to maintain the integrity of our measurements. Then, where applicable, we assess resolution, which tells us how well the instrument can distinguish between two close wavelengths, an important factor for complex analyses. Finally, we have baseline flatness and noise. These parameters help us evaluate the stability of the instrument's baseline signal, ensuring there are no fluctuations or noise that could affect the results. All of these parameters work together to confirm that our instrument is operating within the required specifications, supporting the overall quality and accuracy of our analytical methods. Let's look at this example. The observed peak wavelength is 287.8 nanometers, while the reference value is 287.5 nanometers. To evaluate the accuracy, we calculate the difference by subtracting the reference from the observed value, which gives us plus 0.3 nanometers. Since this difference is within the acceptable range of plus or minus 1 nanometers, we can say it complies with the standards. This is part of our OQ Test 1, which focuses on wavelength accuracy. The standard used for this test is a holmium oxide filter or solution. According to USB acceptance criteria, the difference should stay within plus or minus 1.0 nanometers, or as specified by the manufacturer. The USB reference peaks include 241.1 nanometers, 287.5 nanometers, and 361.5 nanometers. These peaks help us verify instrument accuracy and ensure our measurements are reliable and consistent. Let's take a look at this example. The standard absorbance at 257 nanometers is 0.75, and the observed absorbance we measured is 0.744. To see how close these values are, we perform a simple calculation of the percentage difference. We subtract the standard from the observed, which gives us minus 0.006, then divide that by the standard value of 0.75 and multiply by 100 to get the percentage. This results in approximately minus 0.8%. Since this is within the acceptable range, it indicates that our measurement complies with the quality standards. Now, moving on to the overall photometric accuracy test, or OQ Test 2. We use potassium dichromate in 0.005 molar sulfuric acid as our standard. According to USB, we check absorbance at multiple wavelengths, 235, 257, 313, and 350 nanometers. The acceptance criteria specify that the absorbance should be within plus or minus 0.01 absorbance units or plus or minus 1% of the standard. This ensures our spectrophotometer remains accurate across different wavelengths, which is essential for reliable analytical results throughout the testing process. All right, now let's talk about OQ Test 3, which focuses on stray light. When we're checking this, we have two standard options. One is potassium chloride, or KCl, which we test at 200 nanometers. The other is sodium iodide, Ni, tested at 220 nanometers. The key here is the acceptance criteria established by USB, which is that the absorbance should be 2 or higher. That means, if we measure an absorbance at the specified wavelength, and it's at least 2, we pass the test. For example, if we measure 2.35 at 200 nanometers, that's well above the threshold, so we'd consider that a pass. This test is crucial because stray light can interfere with our measurements, and ensuring the instrument performs correctly at these levels helps us maintain accuracy and reliability in our results. All right, let's look at OQ Test 4, which focuses on resolution. The standard here is quite specific. We're looking at a 0.02% VV toluene in hexane solution. Now, the key criterion set by the USB is that the ratio of absorbance at 269 nanometers to 266 nanometers should be at least 1.5. What does that mean in practical terms? Well, it's about how well we can distinguish toluene from other components based on their absorbance at these two wavelengths. In our example, the absorbance at 269 nanometers is 0.42, and at 266 nanometers, it's 0.26. To find the resolution ratio, we just divide these two values. 0.42 divided by 0.26, which gives us approximately 1.62. And that's the critical point. Since 1.62 is greater than 1.5, this sample complies with the USB criteria. This test is important because it confirms that the separation between these two peaks is sufficient, ensuring the purity and quality of our analyte. It's a straightforward calculation, but it's essential for validating the effectiveness of our analytical method in real-world quality control. Now, let's take a look at OQ Test 5, which focuses on baseline flatness and noise. The procedure involves scanning the baseline from 200 to 400 nanometers using a blank sample. This range is chosen to ensure we accurately capture any irregularities or fluctuations in the baseline signal. The main goal here is to check for stability and consistency in the baseline readings. When it comes to acceptance criteria, we want to see that the noise level is less than or equal to 0.001 absorbance units. That's a very tight limit, but it's important because it indicates a clean and stable system. Additionally, the baseline should be flat without any drift, meaning it stays steady without wandering up or down during the scan. The result we're aiming for is a stable baseline that remains within these limits. When we see a baseline that is flat and stable, it confirms that our instrument is performing well and that the measurements we take downstream will be reliable. Remember, maintaining a stable baseline is crucial for accurate and reproducible results, especially in sensitive analyses. So let's talk about performance qualification, or PQ for short. The main purpose here is to verify that our process actually performs well under real working conditions. It's about making sure that when we put the process into actual use, it still delivers consistent and reliable results. To do this, we analyze a routine sample or a reference standard, something that reflects what we typically work with. Then, we compare the results we get from this analysis with the criteria established by our validated method. This comparison helps us confirm that everything is functioning as expected. Now, the acceptance criteria are pretty straightforward but crucial. We look at the percent RSD, or relative standard deviation, and it should be less than or equal to 2%. This indicates that our results are precise enough. Additionally, the assay result itself must fall within the specified limits. When both these conditions are met, we can confidently say that our process is performing properly under actual working conditions. Overall, PQ is about ensuring that our method is not just theoretically valid, but practically reliable in everyday scenarios, which ties back to our goal of maintaining quality and consistency throughout our operations. Let's take a closer look at the calculation. The mean value here is 0.512, and the standard deviation, or SD, is 0.002. Now, to evaluate the precision of our measurement, we calculate the percentage relative standard deviation, or percent RSD. This is done by dividing the SD by the mean and then multiplying by 100. So we take 0.002 divided by 0.512, which gives us approximately 0.0039, and then multiply that by 100 to get 0.39%. Seeing a percent RSD of 0.39% indicates very good precision, meaning our measurements are consistent. The slide also marks this as a pass, confirming that our method meets the required precision criteria. Now, let's connect this to our overall theme. Accurate and precise measurements are the backbone of reliable analytical methods. This example with the paracetamol reference, standard at 243 nanometers wavelength, shows how we verify that our method performs consistently during performance qualification. It's essential for ensuring quality and reproducibility in our analysis. Let's talk about the frequency of qualification for different activities. First, IQ, or installation qualification, is typically done once after installation or relocation to ensure everything is set up correctly. Then, we have OQ, or operational qualification, which should be performed annually or after major maintenance to confirm the equipment continues to operate as intended. Next is PQ, or performance qualification, which also needs to be done annually or whenever there's a method validation to verify that the process consistently produces quality results. And finally, calibration should be carried out as specified in the SOP, usually every 6 or 12 months, depending on the equipment and regulatory requirements. These schedules are crucial because they help maintain the integrity and reliability of our equipment and processes over time, tying directly into our overall goal of quality assurance. Let's take a moment to review some of the key regulatory references that underpin our analytical practices. First, USB 857 focuses on ultraviolet visible spectroscopy, which is a fundamental technique for identifying and quantifying substances. It's important because it provides standardized methods to ensure consistency and accuracy. Next, USB 1058 covers Analytical Instrument Qualification, or AIQ. This is all about making sure our instruments are properly calibrated and functioning correctly before we start any testing, so we can trust our results. Then there's ICH-Q2, R2, which deals with the validation of analytical procedures. Validation is crucial because it confirms our methods are reliable, precise, and suitable for their intended purpose. Moving to the EU GMP Annex 15, this provides guidance on qualification and validation within the European Good Manufacturing Practice Framework, emphasizing the importance of a thorough, documented approach to validation activities. Finally, the WHO TRS 1010 and 1033 publications serve as international references, offering guidance on quality assurance and validation practices that support global regulatory compliance. All of these references form the backbone of our compliance framework, helping us ensure that our analytical procedures are robust, reliable, and meet the highest standards. They're interconnected, reinforcing the importance of validated methods and qualified instruments in maintaining product quality and safety across the board. For further support, please don't hesitate to reach out. We're here to help you implement what we've discussed today and answer any questions you might have. Just contact us through the provided channels, and we'll be glad to assist. And with that, I want to thank you all for your time and attention. Your engagement is what makes this effort meaningful, and I look forward to seeing how you apply these insights moving forward. Thank you again.